This is a fairly light introduction to data integration. I've given this talk a couple of times to different groups of either trainees or students who are interested in doing some data integration. They're often from a biological background. And what I want to try and do in this talk is just give a general idea of why you might want to do it, and some of the challenges or pitfalls that can easily creep up on you as you start to integrate biological data and try and draw some kind of conclusions or meaning or base further experimentation on what you see. So let's just think about why you want to do it first of all, why the people end up in the classroom listening to this talk. I think the promise of doing data integration pretty much fits with the old idea of growing oak trees from acorns. So starting with something small like an acorn and growing something much bigger like an oak tree. And the hope in going from small to big is that by gathering in all that other information, all that other data together, if we do integrate it in the right way, we end up with something which is much more meaningful, that we can explore and ask questions of and get more value from than the small amount of data that perhaps we started with. But this metaphor of oak trees to acorns gives me the first opportunity really to make one of my key points about doing data integration and the challenges of doing it. And that is, if we think about oak trees, the first challenge is, which oak tree do you mean? There are about 600 species of oak trees. So depending on which acorn you plant will depend on which tree you get. And so what we need to ensure when you start out on the journey of data integration is that you understand the data that you have and that you are beginning with. Otherwise, what you're going to end up with may well not be what you were expecting, or something that will give you answers to the questions that you want to ask of it. So we'll make that a take-home message. First one of the talk. You need to understand the limitations and the context of the data that you start with if you're going to be able to get something useful from a larger integrated data set. Integrating data together does not necessarily mean that it's going to be more informative or give you an answer which will help you. It all depends on what you bring into the mix and what you start with. So let's think about the integration, and we're going to change from trees to butter first. Here we've got some butter. I'm using the butter as an example of an ingredient. This is going to be a cookery example. So in this case, imagine that butter perhaps is a protein of interest that you found in your experiment. From this protein, you want to integrate some other data together and do some predictive biology. So we'll take the butter, we'll get some other ingredients to go with it. We'll get some sugar, some eggs, and some flour. And at this point, I would often ask the class what you can make from butter and flour and eggs and sugar. Sometimes that question meets blank faces, sometimes there's some really enthusiastic bakers in the room and they'll come up with the answer straight away. I can't ask you, I'm going to show you. What can you make from this? Well, first of all, there's one other thing that we might need in this mixture, and that is a bioinformatician. I'm not really trying to insult bioinformaticians here by using the Swedish chef Muppet to show them. It has good comedic value. And it also kind of epitomises, I think, some of the challenges that biologists see of having to talk to bioinformaticians occasionally, because if you know this particular character, you know he doesn't talk anything really intelligible. But actually there's a useful point here. We need a person to mix this stuff together. It may be you, it may be a bioinformatician. We're going to talk about the value of collaboration perhaps in a minute. So we've got our ingredients, we're going to mix them together, we're going to do some cookery. and we will end up with, hopefully, a cake. Well done, anyone who said a cake. Now look at the cake. There's something else in this picture which wasn't in my list of ingredients that's crept in. It's quite important. It's the jam and the cream. Make a nice sponge cake. What goes in the middle? Pretty much perfectly, jam and cream. So perhaps that's our example of predictive biology. We mix together these ingredients. We start with the butter, which was our piece of data. We integrate some other data with that, and from it we would extrapolate that we also want jam and cream. So let's take a step back and think about the ingredients. Same ingredients, but if we mix them together in a different way, do we get something different? Oh, well done if you come up with the answer scones. We can make the same ingredients come together in a different way and produce something that's different to a cake. It's just as nice as a cake, but it is different to a cake. But perhaps the important thing here again is to recognise the other ingredients that have crept into this picture. Scones go brilliantly, traditionally in fact, with jam and cream. So even though we've mixed the same ingredients together in different ways, which we could say would be a problem, what we've actually managed to do is the same predictive biology from that mixture. We have got cakes and scones which are different, but if we're interested in what that leads us to, in both cases this is jam and cream, so it's not necessarily problematic, but we do need to think about how we mix things together. Okay, let's go back to the butter for a minute. Let's say we have selected this as our protein of interest by reading some papers or looking at some data, and let's say we had a description of what was important. It's something that comes from a cow, it contains a lot of fat, that kind of description. We might pick butter, but equally we could say a description like that. We could pick milk. So in this case, I'm using milk and butter as if they're perhaps different isoforms of the same protein. Maybe butter is the protein that's expressed in kidneys and milk is the protein that's expressed in liver or something. But we've ended up picking a different one. Now if we take our same other data sets, we take the eggs and the sugar and the flour, but instead of mixing in butter, we mix in milk. What do we get? We get pancakes. Now we can probably all agree that people who like cake and like scones probably like pancakes too. That's fine. But the message here really is to look at the other ingredients in this picture of pancakes. We haven't got jam and cream. I'm sure you can have jam and cream with pancakes, but many people would choose not to. Maple syrup or syrup is what we would pick to go with pancakes most of the time. So by changing the starting ingredients slightly, even though we've done everything else pretty much the same, we've ended up with pancakes, and the result of that is to have a different outcome in our predictive biology step. So we haven't got jam and cream, we've got maple syrup. Okay, so a slightly silly example maybe, but it illustrates, I think, the key point of firstly understanding what you put in, but also that you can get different things when you mix data together in different ways. And it leads us to the next key point of the talk. Even if you have the same data to work with, integrating it in different ways can give a completely different view of the bigger biological picture at the end. So let's think about the Swedish chef there, the bioinformatician. I said sometimes collaboration is important. Collaboration is often important because people bring different perspectives or skills to the mix. And one of the challenges in embarking on a data integration exercise is having the skills in order to do the data integration. So we can get ideas about that by maybe reading other people's papers and looking at what they've done. Here's a picture of the cake again. We read a paper. These researchers do a data collection, data integration exercise. They produce this lovely cake. And from that they do some interesting biology, and you think maybe you want to do the same thing too. You know all the data, you know all the methods. But if you don't have quite the same skills, you're not going to end up with quite the same cake. Aim for the sheep, you get whatever that is, I'm not sure, but it's not the sheep. Nice minion, not quite the same minion. The things on the left are done by people that, certainly in the cake making world, we would agree have some expertise and skills. And the ones on the right are examples of copies of those where the person doing it didn't have the necessary skills. And whilst these are amusing, this is sort of a significant problem when we look at people trying to do bioinformatics techniques from things they read in papers. Sometimes what's reported in the paper isn't the whole methodology or the whole truth. That's a bit of a different problem. But even when it's all there and you try and do the same kind of thing but without really understanding quite what you're doing, you haven't got the skills, the end result maybe isn't going to be the same. With these cakes, it's quite obvious that the end result isn't the same. The problem is when we start to integrate biological data and the picture is quite complex, it's not always apparent that the end result is not right, and it can be interpreted incorrectly. So I think it's always important to at least consult, a statistician, bioinformaticians, recognise the people with the skills. You can go and get training, and there's a huge amount of great training available to learn key skills. But it is also important to collaborate and work with people who know exactly what they're doing, or at least can spot when the outcome doesn't quite make sense. So that's the other next take-home message. Even if you understand the data and you've got a good knowledge of the biology, integrating data without the right skills to do it can give a poor result or a false impression of the reality of the biological picture that you're trying to explore. So let's change tack slightly and think about bigger pictures. No more cooking. This we're going to look at now using the London Underground map. This is a little piece of it. Hopefully you've spotted this is the London Underground map straight away. What I'm thinking of here is the stations. The big circles here are perhaps proteins and the lines represent interactions or biological reactions, a pathway. And what we're interested in doing is understanding how you go from Holborn to Oxford Circus. And if this is the data we've collected from our experiments, we've got 4 big station data points here and some connections between them, then we've got a pretty simple view of how to get from one to the other. One intermediary step, 2 reactions, 2 pathways maybe. It's quite a straightforward biological scenario, Holborn to Oxford Circus. But I think we all know the map's a little bit more complicated than that, so let's add on a bit more information. Here we've got some other tube lines and some more stations. And immediately we can see that although our original view of Holborn to Oxford Circus via, we now know, Tottenham Court Road is a clear route, it's not the only route. The other routes are longer. We can go Holborn, Covent Garden, Leicester Square, Piccadilly Circus, Oxford Circus, or we can even continue on past Piccadilly Circus and come back up the light blue line or the grey line. But even though the shorter one was what we had at first, it doesn't mean the other ones, because they're longer, are not real. They could represent alternative pathways. They could represent different pathways that occur in different tissues because different genes are expressed. It may be that Tottenham Court Road, as a protein or a gene, is not expressed in certain tissues, and we've got no choice but to go the other route. So as we start to layer on a bit more information, we can see that actually the reality of the situation, the biology that's going on, maybe is a bit more complex, and we need to think about that. Let's see the whole map. There's an awful lot going on here. Whole bigger picture. Many more alternative routes, some quite convoluted and completely different to our original scenario. But can we really say, by looking at it, that they are not true? Having said that, this is a nice visualization of a complex set of data. The map was designed specifically to help people navigate from sort of point A to point B using the underground system. And for that purpose, it's been quite good. It's been around for a very long time, and many of us recognise this. If you live in London, you end up knowing it off by heart. But how much reality is in this picture? How much reality is in this data visualization? Well, let's put this first overlay on. The first overlay highlights which bits of the London Underground map are actually underground. We all know it as the underground map, and therefore it would be an easy assumption to make that this is all underground, but it's not. And that's perhaps one first little highlight, that we can jump to conclusions or make assumptions about things based on how the information is presented to us. And we should always be aware of that. Does it make a difference to how we understand or plan our travel from one point on this map to another? Probably not. But if we start to think about this in other ways, and the fact of whether it's underground or not becomes an important question, the map doesn't actually tell us that, and we might be led down the wrong route. If we expand this out a bit further, I've added on some extra information, and that's train lines. So the London Underground is a type of train, and we've added on some different types of train. And we can see there are other alternative routes appearing, other ways to get in and out of the periphery of our network we're interested in the middle. There's even some shortcuts across it that we could take without going on underground trains, and we could jump straight across the map. From a biological perspective, those kind of shortcuts would be really important, and we could completely change our understanding of what's going on. But if we don't put them in the picture, we wouldn't know they were there. Let's switch to this view. This is the centre of the map again, but this time I've found some extra information to put on this map, and you can see these little numbers all over it. Let's focus in on the bit that we were talking about originally. The red line, Holborn to Oxford Circus via Tottenham Court Road. Now what the little numbers represent are the physical distance between those two stations in, I think, miles. So if you look at this, the distance, the red line between Tottenham Court Road and Holborn is much shorter than the red line between Oxford Circus and Tottenham Court Road. But if we look at the actual physical distance, the shorter one, Holborn to Tottenham Court Road, is actually almost twice as far. If you had to walk this above ground, you walk twice as far to go from Tottenham Court Road to Holborn as you would from Tottenham Court Road to Oxford Circus. Similarly, look the other side of Holborn, where we're heading out towards St Paul's via Chancery Lane. Chancery Lane looks like it's right between, equidistant between St Paul's and Holborn, but actually it's very different. 0.26 miles to Holborn, 0.63 miles to St Paul's. And if you spend any time in central London, you will quickly learn that sometimes it's much quicker to walk between these places than it is to bother going down the tube, getting on the escalators, getting on the train, coming up the other side. The representation of the data in terms of our visualization of tube stations is very good. This map as a representation of the geography of London focused around the tube stations is not very good at all. And if we look at a map of the geography of the tube, we can see that actually it bears no resemblance at all to the sort of graphic that we're used to seeing. And also, if we go back to the middle of the map, here I've added on bus routes. So if our question is how to get from point A to point B using the underground, the original map was quite good. If it's how to get from point A to point B walking, then the London Underground map we've already seen isn't so helpful because the distances are wrong. If we don't think about trains at all and we think about buses, then we've got a completely different picture. So this is really bringing us to a take-home message about visualising the complex picture of the data. How and which data is represented has a significant effect on our understanding. It helps us in some areas, but it can misrepresent the truth in others. Now, that misrepresentation of the truth isn't necessarily an intentional misrepresentation by the people who create the figure. It's about how we interpret the figure and try to apply it to our own questions or our own interests. And we have to remember, when data is presented in papers, it's often presented in a way that aids our understanding of the key concepts or the conclusions or the meaning that the authors of the paper want to extract from it to present to us. And we can explore around their meaning and their interpretation, but it doesn't necessarily mean that that is going to help us with our questions. And that's why it's quite valuable as bioinformaticians to always be able to have access to the raw data in the supplementary materials or put into a repository so we can come and analyse it and do our own visualisations. So we've got to think about why a visualisation was produced and what data has gone into it before we start interpreting it for our own use, and make sure our use and understanding of it is aligned with the original purpose of the visualisation. So again, let's switch. We've gone from acorns to cookery to the London Underground map, and now we are on the moon. So this example is one that I've taken from this controversy, if you will, the conspiracy theory over whether or not the moon landings actually happened on the moon or in a studio. I think they're on the moon. Some people think they're in a studio. Fine. But where does that come from? And one of the key pieces of evidence, if you will, as to why people think that maybe the moon landings were not true and did happen in a studio is a few photos like this one. This is one of the key photos. So this shows one of the astronauts coming down from the lunar lander. And the reason this is held up as being a problem is that he's quite well lit and he should be in shadow. He should be in the shadow of the lander, but he's quite well lit. And the argument here is that he could only be that well lit if there was another light source, as if he was in a studio with studio lighting. So a piece of research or study that interested me the last couple of years that I put in here comes from NVIDIA, the graphics card manufacturer, and they'd released a new graphics card that they were showing off its light rendering capabilities, and they took on this as a sort of challenge, as an example to try and show off the capabilities of this card. So what they wanted to do was try and explore the lighting in this picture. Could they render the scene, and by rendering this scene, understand the lighting arrangements within it, and you know, perhaps not purposefully try and support the conspiracy theories, but come up with some evidence or answer to this question about the picture. So this is an image taken from their rendering exercise. It's the same. It's a photo from the same series of photos as the previous one. We've got the astronaut here, and he has been rendered in software hardware by NVIDIA. And what we can see from this is actually he's quite dark. Now they tried to put into the picture as much of the environment as they could, so they've rendered the reflectiveness of the lunar surface and the metallic parts of the lander and the position of the sun in the sky and stars and things like that. That anything that might have an impact on the lighting in this render, they were trying to demonstrate lighting capability of the render, so they put it all in. But what we've ended up with is an astronaut who's quite dark, and on the face of it, that begins to support the ideas of the conspiracy theorists that this needs another light source. Now their step forward in that is it needs another light source, it must be studio lighting. If you come back a step and say, well, he's not bright enough, it needs another light source, what's missing from this environment? And you take that step back and you think about what's here, which is what the researchers at NVIDIA did. they realised that there was something missing from the environment. The thing that's missing from the environment is the person who took the photograph. So the other astronaut stood on the lunar surface in a bright white spacesuit, standing in the sun, taking a photograph of the person coming down the ladder. Here he is in the picture. So he will potentially be acting as a reflective light source. It doesn't have to be a studio lamp. It could be the other astronaut. So when he gets plugged into the render and the scene gets re-rendered, we go from this, the scene without the other astronaut, to this, the scene rendered with the other astronaut. And I'll just flick backwards and forwards for a second. Without the astronaut, with the astronaut. Without, with, without, with. We can see there's a brightness in the second scene. We're getting reflected light from the other astronaut. It's lighting the scene and actually filling in the blank of where is this other light source. We get these two side by side, in fact. We get the original photo from the lunar mission on the right, and we get the render from NVIDIA on the left. We can see they're actually in a reasonable amount of agreement. This is both a lesson in thinking about the bigger picture and the missing data, but also a good advertisement for NVIDIA's graphics card capabilities. There you go, fantastic picture. So this brings us to the last of the major points about data integration. We don't usually, in fact hardly ever in biology, have all of the data, all of the information in our environment or scene that we're trying to look at. And unless we recognise that and keep that in mind, we can be led to false conclusions, which in my opinion the conspiracy theories are. You know, there's always other things that we cannot have studied in our experiments, or we don't have the data for, or we haven't got complete data, or we don't know how it works or integrates. And so our integrated data set gives us perhaps a nod in the right direction of some interesting biology or something to explore. But we've got to be careful what assumptions we put on that and the decisions we make from it, and make sure we're not missing something or discounting something that we should have thought about. So as a kind of conclusion to this talk, there's some key things in amongst the fun examples that I want you to take away from that, the things on the black slides. As a kind of final comment, I kind of just want to reiterate, there is a huge amount of potential in big data, bioinformatics, data integration. It's all kind of buzzwords and hot topics and that kind of thing. There's a lot of potential in this to help us understand biology better, maximise our investments in public data spending, in small grants in labs that just have limited resources that can get one or two results and then plug that into this bigger picture. There's a lot of potential for it. But, and this is where my opinion creeps in, and I hope most people would agree with me, but some of you might not. I would certainly say it will never provide an absolute answer. Bioinformatics generally does not provide an answer. Results from bioinformatics analyses, experiments, data integration, etc. should be used to guide further experimentation, certainly at least validation of what you're seeing in the in silico experiment. But guide further experimentation and exploration, use it to generate new hypotheses and questions to go and look at in detail. It's kind of a cycle process. You generate a little bit of data, do some data integration, go find some ideas, go back to the lab, do some more experiments, get a bit more data, maybe do some more data integration. Hand in hand, biologists, bioinformaticians, labs, computing, all together, working together to try and answer these questions. But you can't really do it alone, and you've got to collaborate. And you've got to keep in mind the challenges in the data integration, the bioinformatics, as much as you would think about difficulties in an experimental setup or validation of a particular antibody or preparation of a buffer. It's still science, it still needs a critical eye and thought behind it, and designing your experiments and questions appropriately. So that's the end of the talk. I hope it's been somewhat useful and at least given you some ideas to think about. I'm always happy to enter into discussion online. We often have good conversations in the classroom after this, but obviously that's not helpful here. If you want to drop me a line, have a discussion. If you completely disagree with me, then that's entirely fine too. But get in touch. Okay, I hope it was helpful. Thank you for the time listening.